Well, Gabe, thank you very much indeed for that very good session. It was a great session. Excellent speakers, wonderful discussions. So now we are going to be moving on to part 3 of the conference. And this will be the Fireside Chat. And this is the Fireside Chat on knowledge and resource sharing. So we have a group of 6 speakers, 3 of them from universities, and the other 3 are NIH speakers. The 3 speakers from the universities will be telling us about some of their exciting research that are relevant to the conference we are having here. And our NIH colleagues will be sharing with us some information about the work that NIH is doing that is also relevant to conference here. So we have speakers today, Dr. Greg Talavera from San Diego State University, Dr. Raul Cateno from the UT Southwestern Medical Center, Dr. V. Wendy Setiawan from Keck School of Medicine, and we have from NIH, Doctors Reena Das from the National Institute of Minority Health and Health Disparities, Dr. Damali Martin from the National Institute of Aging, as well as Dr. Yuling Hong from the National Heart, Lung, and Blood Institute. If you're interested in knowing more about these speakers, I would recommend that you please consult the speakers' bios, which have been published on the conference website. So we will keep the questions for the end of this session today, in this session that we're having currently. But I would also like to encourage participants to please feel free to post your questions in the chat box. And also, if you have questions, please raise your hands and I will try my best to accommodate your question. I just want to say that I am aware that we have several people who are listening in with lots of expertise. So please do not be shy to ask questions or share opinions. So we'll really appreciate that. Okay, so our first speaker today will be Dr. Greg Talavera. Dr. Greg, over to you.
Thank you. First of all, I'd like to thank the organizing committee to invite me, and in particular Tram, who convinced me that a cardiovascular researcher had something to contribute to this NCI conference on cancer. So I'm going to talk about what appears to be a very relevant topic that we have learned and gained from the Hispanic Community Health Study of Latinos, which is an ongoing cohort study. So next slide. So the Hispanic Community Health Study is a longitudinal cohort which was designed to identify the prevalence of cardiovascular and pulmonary disease in a cohort of US Hispanic Latinos of diverse heritage, and indirectly also explore the protective and harmful factors that might be contributing to the Hispanic paradox of cardiovascular disease. So these are the sites where the study was conducted: San Diego, Miami, the Bronx, and Chicago. We followed a similar protocol. Data was captured at the coordinating center in University of Chapel Hill, and John Kuntz is our current project officer. This is an overview of the timeline. The initial baseline recruitment and assessments took place from 2008 to 2011, and then the second and third visit. My remarks and comments are going to be focused on our baseline experience recruiting and interacting and engaging in our sample. This is the results of the recruitment. We ultimately ended up with 16,000 individuals, and this is their distribution by what we call Latino heritage or background. The major group being Mexican, followed by Cuban and Puerto Rican, with other individuals, other background groups. This is what the sample looks like at the various sites. San Diego, predominantly Mexican, as well as Chicago, and in Miami, of course, predominantly a Cuban background, and in the Bronx, a Dominican background, which was somewhat of a surprise to us when we started recruitment. I wanted to share this because there's been a lot of discussion about Hispanic heritage or background in this conference, and I wanted to share what we ended up with in terms of genetic ancestry. These are estimates of continental ancestry proportions on the autosomes of the individuals that agreed to have their genetics identified. And principal component analysis identified 3 major gene pools: the European, Amerindian, and African. And here, which represents what the self-identified heritage group was, Cuban, Dominican, Puerto Rican, and so forth, their composition overall by these 3 gene pools. And as you can see, there's these 3 on the right side, Mexican, Central American, South American, have relatively small amounts of African contribution, whereas the Caribbean group has a more significant contribution from that. So it's interesting to keep in mind, because, you know, when we study any cancer or heart disease issue, there's always a genetic component, and this is useful information for us. So with that said, as a background, I wanted to talk about some of the, well, basically now, the experiences that we have and the perceptions that we have as researchers, my colleagues across the 4 sites, about how to approach the sample that we have. And this may serve as an idea or a methodology that could be replicated in future research going forward. But we chose the Hispanic Latino identifier because we wanted to be able to compare our results with national studies and with the US Census, which is the question they ask on the US Census. Doesn't mean that national origins or ancestries or other questions are not useful, but I'll talk a little bit more about that. And then we also discovered over time that you know, specific terms that refer to place of birth can also be very important, depending on which term you use and how you ask it and so forth. So for example, in terms of place of birth, we decided to use born in the US mainland or born in the US 50 states when we were interviewing our participants and when we published. And another point that we wanted to capture was immigration status, and we tried to parcel out first generation or immigrants from second and third generation. So our group, our sample is really 80% immigrant population with 20% second and third generation, quote unquote, Latino Hispanics. There's been a lot said already on language use in the study, so I won't go into that. People ask us, why don't you use the Latinx term when defining our population? Well, it's a relatively new term, and when we, as I'll tell you later, we struggled just to get Latinos to identify as Hispanic Latinos when we did our report. So I may be behind time because of the mix-up here. So I'll just point out that we do have a question that deals with immigrant status, and it's broken into 2 categories with these response options, and I'll let folks review that at their leisure. So again, just to remind folks, the term Hispanic Latino and other definitions that people have used are constructs. But the thing that we discovered is that people who immigrate from Latin America really don't understand those terms when you approach them for participation in research. There are many of our participants who we knocked on their door and tried to recruit them into the study were puzzled by those terms. But when the staff were noticing that they spoke Spanish and they were in a high density Spanish Latino area, we allowed them to ask a second question and tell them that the purpose of this study was really to address people from Latin America. And then they got open to the idea of being labeled as Hispanic or Latino. When it comes to race, as many people have alluded to in this conference, it's a difficult term. So after we determined that they were Hispanic Latino, and we tried to ask the race question, there was more confusion, and only 25% agreed to answer that question, and the rest refused or simply didn't really understand it. So that can happen. And this is a reminder that our population were adults, 18 to 72 at the time of recruitment. So this is the strategy we used at the door. Do you consider yourself to be of Hispanic or Latino background? And if they said no, recruiters were allowed to ask the second question about being from Latin America. What our recruiters were telling us was that they mostly thought that they were Puerto Ricanos or Nicaraguense and Mexicanos. And this has been identified in some of the presentations of this conference. So after we got them to agree to be Hispanic Latino and got to know them during the interview period, we also asked them what their heritage was. And we showed them a list of Latin American countries. And that's how we determined a Hispanic Latino heritage for our sample. So just a few recommendations. We recommend the term heritage or background as a question and as a way to describe the subpopulations that we're dealing with. And our English majors have told us not to use the word subgroups because that suggests a hierarchical relationship. So we always recommend to our manuscript writers to use subpopulations and not subgroups. I have already mentioned that 20% were US born, so you can't label in your manuscripts or in your presentations that all you had was Mexicans or all you had was Puerto Ricans. You have to use the adjective heritage. And just to give you an example how this plays out in cardiovascular risk factors, we have the risk factors on the left and the heritage groups on the top row, and the red indicates the highest and the green indicates the lowest. And you can see that there is quite a bit of diversity and risk factor profiles for Hispanic heritage groups. And then the other thing that has surfaced as probably our best proxy for acculturation, which is length of residence in the United States. This is a relationship between prevalence of diabetes and length of residence in the United States compared to the US born. So this is how our lexicon of terminology has resulted in ways of presenting the data by diverse background. And just to let you know that at our site, we have all our measurement instruments and a lot of our recommendations that I presented today. And I apologize for going over, and I'll stop there.
Great. Thank you, Greg. I'm sorry about the technical issues there.
Yeah, I learned something today.
Okay. So our next speaker is Dr. Raul Cateno, and he will be talking about the Hispanic American baseline alcohol study, some of the lessons and experiences learned there. Okay, Dr. Raul.
I'm unmuting myself here. Okay, there we go. All right. I'm not going to focus only in the Hispanic Americans baseline alcohol survey, but in other surveys that I have done as well to give you a more comprehensive idea of the experience that I have gathered in doing alcohol research and epidemiological studies with Hispanics in the US. I have a collaborator in this presentation, Dr. Patrice Veit, who has worked with me for almost 20 years now. Okay, so basically, I'm going to talk a little bit about the study characteristics that I have done, and I'm going to highlight a few methodological points as you see in the slide. Sample and data collection of these studies, interview language, the issue of sensitive data collection, given that I work in the alcohol field, and the issues related to field work. Next slide, please. As you see in there, there are basically several national studies and another more regional studies that I have done. Before the Hispanic Americans Baseline Alcohol Survey, I did 4 national surveys, which oversampled Hispanics and Blacks. And after Abelas, I did, with my collaborators, study on the US-Mexico border. Then we did another survey in Puerto Rico and are now doing a study in California that includes oversample with or focus on whites and Mexican Americans. And on a whole, if you add the interviews that we've done in all these surveys, it's over 13,000 interviews with Hispanics, most of them, but also some whites and Blacks. Next slide, please. All studies, with only one exception, the California border study that is ongoing, were conducted with general population samples of the household population, the adult population in the United States, with face-to-face interviews. The data collection in the California border study that is ongoing was both on the phone and online, and a considerable number of participants, as you can see from the slide, 86 participants opted to be interviewed online. Now, this is a young, relatively young group, 18 to 39. So maybe that's why we had so many of them able to join online and be comfortable with that mode of interview. Usually what we do is we assess Hispanic ethnicity based on self-identification, but we also ask questions about place of birth, race, place of birth of parents, and if the person was born outside the United States, age of arrival. Next slide, please. The issue of sensitive data collection and minimizing underreporting. In the alcohol field, we deal with approximately 40 to 50% of underreporting of alcohol consumption in the United States. The same things happen, of course, when you ask for other illicit drugs like marijuana is in California now, or illicit drugs. So it's very important to establish rapport and then follow with questions about sensitive information. When we have to do that, and I imagine that some of you at some point in time may want to ask about alcohol consumption, because after all, alcohol is a carcinogenic, we usually start with frequency questions at a very high level. And we do that just to avoid, again, underreporting. So if you want to know about frequency during a week, you may ask, for instance, please select the following frequency categories. And the first one is, let's say, 20 to 30 times a week. Most respondents are not going to drink that level, but the respondents that do, when they see a high frequency as an option in a question, there is more of a tendency to select that question than otherwise. We do the same with quantity. I would strongly avoid the use of adjectives and words like average, usual, typical, because what happens is the following. If I am a drinker and I have 2 drinks per day, 5 days a week, and then 2 days a week, instead of having 2, I have 10. If you ask me what is my usual drinking pattern, I may say to you, well, the usual or the average or the typical is 2 drinks per day. And I will not inform you about the 2 days in which I may have 5 or 6 or 7 drinks. And with that, not only I am underreporting my consumption, but I'm also not giving you information about important risks that I have, because in 2 days a week, I have my alcohol consumption, take my blood alcohol level above the legal limit, and therefore that brings special risks to myself and those around me. Next slide, please. Of course, if you're interviewing Hispanics, you're going to have bilingual interviewers, Spanish and English. The Abela study was a study that was designed specifically to interview Hispanics of several national groups in the United States. We were expecting to interview about 1,500 people in each of the 4 major groups: Cuban Americans, Puerto Ricans, Mexican Americans, and South Central Americans. So we went through an elaborate process of translating and retranslating the questionnaire with a group of interviewers from several countries in Latin America to assemble the questionnaire. And we also created a crosswalk of terms, focusing on terms that were used more in some areas of Latin America than others. Well, the crosswalk was hardly ever, if ever, used during the interviews. The standard Spanish questionnaire that we use was acceptable to more than 90% of the respondents, maybe all respondents, and we had no problems at all with interviewing in Spanish, in standard Spanish. Interviews in Spanish usually last a little longer, and you have to take that into consideration too, because the time of interview is one of the ways that survey research work, field work, charges for collecting data. Next slide, please. So data collection is very expensive, especially if you're doing face-to-face interviews. And if you're going to do that, you're going to have to hire a very reputable agency, get a lot of background information, and participate actively in training the fieldwork personnel. This is an area where you cannot really cut corners. Problems do happen, and I'm going to tell you about 2 problems that I have faced. The first one was in the Abela study, where because the interviewers were not very well selected, there was considerable cheating. In other words, interviewers submitting questionnaires that did not come from the respondents were filled by themselves. This problem was relatively widespread to the extent that the survey had been planned for 6,000 interviews, and I could only have 5,200 because the rest of the interviews I had to throw away and or use money to correct the problem of interviewing. In the study that we have now in California, we had a 7% response rate for the survey in Imperial County because of issues related to COVID, and also we think immigration policies affecting those on the border. There was no cooperation with the survey there. And of course, the interview mode is a battle against time because you have a lot of data that you want to collect, and at the same time, you have a finite time to interview respondents. Next slide, please. And the last one. All right, thank you very much. That's what I had.
Thank you very much for an excellent presentation. We'll move on. Our next speaker is Dr. V. Wendy Setiawan, and she will be speaking to us about the multi-ethnic cohort study.
Can you guys hear me okay?
Yes, we can.
All right, so thanks, Dad, and thanks for the organizers. Good morning, good afternoon, everybody. It's my pleasure to be part of this important workshop and to represent the multi-ethnic cohort study. So I will begin my presentation with some backgrounds about the MAC and how this cohort's unique resources have provided great opportunities for epi research, especially in minority populations. And then I will talk about some challenges and lessons learned from working in the MAC with a special emphasis on the Latino populations. Okay, let me see if I have a... All right. So the MAC is a collaborative, multi-institutional and well-integrated program of epi research aimed at identifying environmental and biological risk factors for cancer and other chronic diseases, assessing contributions of these risk factors to ethnic racial health disparities, enabling translational research addressing health inequities, and contributing ethnic, racial, minority data and or samples to large collaborative studies. So the MAC is a prospective cohort study established between 1993 and 1996, jointly by Larry Colonel in Hawaii and Brian Henderson in LA. The cohort consists of more than 215,000 individuals with these 5 major ethnic groups, including Native Hawaiian, Japanese American whites, African Americans, and Latinos. So the Latinos in the MAC, they're mainly from the LA County. Diet and lifestyle factors were collected at baseline, and we also collected biospecimen from about 70,000 participants. So participants are followed actively using follow-up questionnaires about every 5 years to collect updated data on smoking, diet, other lifestyle factors, medical history, screening behaviors. And in the last 2 questionnaires, knowing that the MAC is aging, we collected data on physical functioning, quality of life, and also social and emotional support. For outcome ascertainment, we link the MAC participants to cancer registries to obtain cancer incidence and to state and national death index to ascertain death. In addition to this registry, we have also linkages to Medicare and California hospital discharge data, which have allowed us to study lots of non-cancer phenotypes now in the cohort. So as I mentioned briefly, we collected biospecimen in the MAC. In the mid-90s, we focused on selected cancers. In early 2000s, we did prospective collections of blood and urine from about 70,000 participants. Since then, we also collected saliva samples, tissue blocks from cancer cases, and a PO1 study within the MAC led by Loic Lemarchand also collected stool samples. So biospecimen in the MAC has contributed to numerous genetic susceptibility and biomarker studies of cancer and other chronic diseases. So overall, we've been very blessed with the rich resources of the MAC to study multi-level drivers on many diseases, looking at genetics, lifestyles, disease characteristics, and also environmental exposures in multiple ethnic groups, including Latinos. So here is the MAC composition. So for the rest of this presentation, I will focus on the Latino group, which is the 3rd largest group in the cohort. We have more than 47,000 Latino men and women in the cohort, and about 18,000 of them have biospecimen. Looking at the Latinos, the majority of MAC Latinos are US born, have you seen here. Majority of foreign born are from Mexico, about 35%, and about 12% are from Central and South America. Overall, if we look at the MAC Latinos, 68 to 70% are of Mexico origin. In terms of looking at years living in the United States, more than 75% of MAC Latinos have lived in the US for a long time, more than 26 years. This is at the time of cohort entry. And only 5% of them have lived in the US for less than 10 years. The table here shows the number of incident cancer cases accrued among MAC Latinos since the cohort inception. Overall, there are more than 96 100 cases. And the highest number of cases are observed among the top 4 cancers: breast, prostate, colorectal, and lung. And I also provided the number of cases here with biospecimen and also those with pre-diagnostic blood samples, which are extremely valuable for biomarker studies. So we have many exciting examples of studies in the MAC, but I will just highlight a few of the recent ones focusing on Hispanics. So leveraging the information about birthplace of the participants and of their parents, we group Mexican Latinos into immigrants born in Mexico, US borns with both parents born in Mexico, US born with one parent born in Mexico, one parent born in the US born with both parents born in the United States. And this is just a comparison. This is the non-Hispanic whites. So what we've seen in this graph here, we found that both birthplace and increasing generations are associated with increased risks of overall cancer mortality. And the risk of cancer death is approaching non-Hispanic whites with increasing generations. For specific cancer and chronic disease, we previously showed that US-born Latinos have high risks of developing liver cancer and died of liver disease among Latinos in the MAC. In terms of risk factors, we found that diabetes is strongly associated with liver cancer incidence. And if you look at across ethnic groups, the highest population attributable risk is observed in Latinos. We also showed that there are ethnic racial differences in underlying etiology for liver disease, with NAFLD being the most important etiology among Latinos. So these findings underscore the importance of liver disease prevention in this population, especially for metabolic syndrome-related conditions like NAFLD. Just an example of genetic study, we utilize GWAS data in the MAC to build polygenic risk score for NAFLD in multi-ethnic populations. And here we show that PRS, polygenic risk score, is associated with NAFLD development and progression in Latinos and other groups. So this shows the utility of PRS for NAFLD risk stratifications in high-risk groups like Latinos. As we can see from all these examples, MAC data are so valuable, providing insights into etiology and also prognostic factors that influence health in Latinos and other minority populations. As of today, the MAC data and/or biospecimen have been shared and used widely in more than 80 institutions. You can see the list there, and then you can see this in the map, and more than 800 papers using MAC data have been published. Now, some challenges specific to Latino participants in the cohort. Now, if you look at the response rate for across different groups in the MAC, this is response rate for follow-up questionnaires. The response rates for participation and returning follow-up questionnaire in Latinos over time are amongst the lowest in the cohort. As we gain insight from our long-term study coordinators and interviewers, we learn several factors that can improve recruitment and maintain participations among our Latino participants. Trust and reassurance of confidentiality are critical during recruitment and data collection. Making sure potential participants understand fully about the research process and its possible benefit beyond information that we include in consent forms are very important. Having bilingual study coordinators and interviewers is a must, and we make sure all our questionnaires and paperwork have Spanish translations. So we found is that if Latino participants find the objective of the research attractive, if they are treated with respect and do not find the research process threatening, and they develop connections with the research staff during interviews on other activities, they are more likely to be more receptive to requests for continued participation in research. Our Latino participants often ask for compensation, so if budget permits, it can be helpful to increase participation. In terms of follow-up, our Latino participants need more calls to remind them that they are part of the study during additional data collection. Now, after the data have been collected, we encounter a different type of challenges. So with country of origin and birthplace information available, we were able to perform stratified analysis by these variables. However, as you can see here, the number of Latinos who are not of Mexico origin is relatively small in the cohort. So when we study certain phenotypes, we usually have to only include the Mexican Latinos, knowing that there are differences in lifestyles and genetics ancestry between Latino subpopulations. And for genetic and biomarker study, despite the large cohort of the MEC, depending on the disease phenotype, sample size in Latino group can be limited, and the challenges have been finding replication studies for our findings. So in this last slide here, I put the MEC website information. Feel free to check it out and find out more about the cohort. And for those who are interested, there's also information about how to access the MEC data. I'm happy to answer questions later toward the end. Thanks.
Thank you, Wendy. Wonderful presentation. Hopefully we'll get some good questions for you at the end. So our next speaker, we are now going to transition to our NIH speakers. So Dr. Rina Das will be our first NIH speaker.
Hi, everybody. Thank you for the invitation to share information about NIMHD programs and few selected funding opportunities that might be of interest. I think this workshop is very timely and of great interest to NIMHD. So here, this slide, just wanted to share the mission of NIMHD. Some people may not be aware of our institute. We are a small institute, and we are focused on promoting research in minority health and health disparities. And we also have a lot of interest in increasing the diversity of the scientific workforce. And we collaborate with many institutes at NIH to promote health disparities research. In terms of the extramural program, here you can see that the NIMHD extramural program is divided into three main divisions. And these are the clinical and health services research, integrative biological and behavioral sciences, and community health and population sciences. So together, these three areas provide a comprehensive platform to advance the science of minority health and health disparities. And there is a great emphasis in increasing the investigator-initiated projects, especially R01s at NIMHD. So this is a research framework that a lot of you may be familiar with, but I really wanted to stress the importance of this framework because of the multi-domain and multi-level influences on the variety of health determinants relevant to understanding and addressing minority health and health disparities. When you look at the portfolio, we see a lot of research has been focused on this first and second box on either biology and behavior at the individual level. But as we know how complex the causes of health disparities are, we think that it's important to look at not only at the individual level, but maybe at the family, community, at the system level, and try to understand how these different multiple factors are linked and how they drive disparities. So in this slide, I just wanted to share that NIMHD funds research for many different population groups. These are multiracial ethnic populations. We also fund research in rural, low socioeconomic status, and sexual gender minority populations. And we are not focused on any one disease. So we do fund research in a variety of different diseases, and cancer is one of our really highly funded area of research. We have also funded research in, besides Hispanic, African American, and other populations, in Asians and American Indian populations also, we have a lot of research in cancer. When you look at the cancer portfolio, when I was looking at the numbers, we have like over 30 or 40 grants, but we have some unique programs where these are funded from. And some of our center projects that I have listed here, the Research Center in Minority Institutions, the RCMI centers, the Center of Excellence, the Transdisciplinary Collaborative Centers. So RCMI, COE, TCCs, these are some acronyms that we have used, and a variety of different of these centers are focused on Latino populations, whether they are in Puerto Rico, Texas, California, or other sites. So the example I wanted to share with you is here is an example of a grant funded, RCMI grant funded in Puerto Rico, where they are trying to integrate social factors with biological factors. And this is an, again, area that we are trying to promote how the social adversities, such as exposure to crime and exposure to physical violence and anxiety, affect immune cell function in breast cancers, in breast tumor environment. This is a study done in Puerto Rican women, and the data here shows that if you're exposed to these social adversities, then there is high rate of these tumor infiltrating cells into the tissues, and this could be some mediators of poor outcomes in these patients. Here I have listed some selected funding opportunity that might be of interest to some of you. These are some funding announcements led by NIMHD, but we partner with NCI and other institutes quite a bit. So some of them I just wanted to highlight. Structural racism and discrimination is an important area of research, how it affects health. We had a workshop, we had a recent RFA. So if you are interested in that topic, you can always submit to the parent R01. We are also interested in immigrant health. This is a funding announcement that we had in collaboration with NCI and other institutes that covers a wide variety of topics, data from some of the Latino subpopulations, the effect of immigrant policies and its health outcome. I've listed other funding announcements that might be of interest, especially on role of work in health disparities and the effect of disasters on health and healthcare. These are some unique funding announcements that might be of interest. Sorry, can I go back? So I just wanted to mention one of the funding announcements on liver disease and cancer disparities. This is, as you have heard, that the incidence and mortality rates are quite high in Latino populations. So I just wanted to mention that we have a really concerted effort in understanding the causes of liver disease disparities in different populations. And we have funded a variety of topic areas in this funding announcement. This is again in collaboration with NCI. And as you have seen data that geography plays a role, Texas seems to have a very high rate of liver cancer rates, and especially South Texas. So we wanted to really promote research where you can look at multidisciplinary research where you combine the social, the biological, the behavioral, other risk factors, and see how it affects these diseases. and does it affect different risk factors? And here I've given you a list of some of these grants that we recently funded in Hispanic population in the border area. And I know we are running out of time. This is another area that we are interested in on social epigenomics, where we are trying to combine the social sciences with biological sciences and see how can we understand the underlying mechanisms of health disparity. And here we have funded several grants on Latina women. There is one on the by Dr. Seawald on genetic damage living with food desert zip codes and how that affects breast cancer in Latino and African American women. So I thought I had a little more time, but anyway, I just wanted to highlight these areas that are of interest to NIMHD, especially multi-level research, multidisciplinary research. I know there was some talk on healthcare models. So here are some list of new research concepts that were approved by our council on integrated care model, healthcare delivery for multiple chronic diseases. We also have one on practice-based clinical research network to improve healthcare for populations that experience health disparities. And I have some list of resources on a book that NIMHD helped publish on health disparity research that has a lot of protocols and approaches that might be of interest. We have a science vision on a journal that we published in 2019. Take a look at some of the scientific recommendations on the research for health disparities. And the Phoenix Toolkit is another resource to look at how some social determinants of health measures can be collected. And I think that's about it. And here is our contact information. If you do want to look at some of the funding opportunities, you can visit our website. And I'll be happy to take any more questions. Thank you.
Thank you, Rena. Thank you very much. We'll be keeping our questions for the end. So our next speaker is Dr. Damali Martin. She is now at the National Institute of Aging. So Damali, over to you.
Good afternoon. I would like to thank the organizers for allowing me to present information on the research support for Hispanic and Latino populations by the Division of Neuroscience at the National Institute on Aging. For this presentation, I will focus on Alzheimer's disease and related dementias, which I will refer to as AD and ADRD going forward. The National Institute on Aging is the leading institute for aging research globally. Our mission is to support and conduct aging research in a number of different areas, and we also seek to foster the development of our researchers in the field. We also provide research resources, and we disseminate information about our advances in research to the public, healthcare professionals, and the scientific community. The Division of Neuroscience, one of the 4 divisions in NIA, has been charged with supporting research that would lead to better understanding of the mechanisms of brain aging in AD and ADRD. The division utilizes a comprehensive approach that allows us to examine the etiology of this disease along a continuum from basic biology to population-level studies, including epidemiology, behavioral research, translational, and clinical research. So I'm presenting data from 2 observational studies of AD and ADRD, which have documented significant racial and ethnic disparities with higher incidence and prevalence among 3 race and ethnic groups, namely African Americans, American Indian and Alaskan Natives, and Hispanic populations. And these disparities held across the 3 different age groups as you can see on your graph on the right. And I wanted to share this reference with you, which describes AD and ADRD in the Hispanic and Latino communities. I will not dive into specifics, but I just wanted to point out that one of the overall take-home messages from this paper, which is we have to pay attention to the diversity of the Hispanic and Latino populations as we design our research studies, because these populations represent an additional challenge since it may be distinct characteristics associated with disease, in this case with AD and other comorbidities. So we recently hosted our 4th NIH AD Summit, which is done every 3 years. And these summits serve as key strategic planning meetings tied to the first goal of the national plan to address Alzheimer's disease. And so we use these summits to develop and update research milestones. I've listed the research milestone that's related to health disparities here on the slide on your left. But what I really want to draw your attention to is to the health disparities framework, which is a great example of joining both biological factors and social determinants of health into precision medicine. This framework allows us to better delineate causal pathways and broaden the scope for tangible targets to address AD and ADRD disparities. It also allows us to consider the levels of intersecting domains and the level of influence and evaluating how those factors influence disease risk across the entire life course. So with this in mind, I would like to touch on a few examples of the types of research support that DN offers on the Hispanic and Latino populations. Our health disparities and AD disease NOCI supports several investigator-initiated research projects, including those on Hispanic populations, and I've listed 2 examples here. One study collects data on Blacks and Latinos from across the US. Another one focuses on Blacks and Latinos within a particular neighborhood in New York. But what is important for both of these studies is that the investigators have both been successful in utilizing the health disparities framework to develop a comprehensive study that would lead to a better understanding of the etiology of the disease. And so they could include the collection of data from across the 4 different domains in the health disparities framework. So the AD sequencing project, or the ADSP, is one of our signature projects. And under this project, we support research to discover long-term treatments for the disease by the identification of risk and protective genes and the underlying molecular pathways. The follow-up study in diverse population aimed to do the exact same thing in our minoritized populations. Specific to Hispanic populations, we are currently sequencing 2,500 cases and 4,500 controls. I've listed a few of the studies that are participating in this sequencing project. We're seeking to increase these numbers, and ultimately our goal is to sequence up to 18,000 Hispanic and Latino populations from various studies. Our AD research centers are pivotal to conducting cutting-edge research for AD, and these are also supported through our division. The ADRCs support clinical and epidemiologic research in several minoritized populations, and they are supported by a number of research infrastructure that supports the collection of various data on our populations, for example, the NAC, or the Standardized and Central AD Neuroimaging, which collects neuroimaging data, and so on. About 8% of the population covered by the ADRCs are Hispanic, and so there is some room for improvement for increasing the number of Hispanics and other minoritized populations in our studies. So one way in which we can do this is through the creation of opportunities to increase diversity recruitment and retention in our research. And this initiative listed on the slide is one example in how we can support these activities. This PAR supports projects which aim to foster a collaborative network of teams to engage large, diverse populations and to accelerate research participant recruitment into aging research and NIA-funded clinical studies. And 6 out of 10 grants funded under this PAR focuses on Hispanic populations, 2 exclusively on Hispanic populations. And in this last slide, I just wanted to share that approximately 19% of our grants in our research portfolio related to medically underserved populations are focused on Hispanic populations, either solely on Hispanics or comparing Hispanics to other populations. And so as we continue to evaluate our progress, we will seek opportunities and resources to address scientific gaps. And we also look forward to working with our NIH colleagues. And I would like to thank you for your time and attention.
Thank you very much, Damali. Our next speaker is Dr. Yuling Han.
Good afternoon or good morning, depending on where you live. And first of all, many thanks to NCI for hosting this timely, important workshop, and to Dr. Lam for the invitation. Over the next few minutes, I'm going to share a few points on how the Division of Cardiovascular Sciences, especially the epidemiology branch, implements the NHBI's strategic vision to support Hispanic health research. NHBI launched the listened strategic visioning process in 2016. We incorporate the suggestion from all the stakeholders, including the research investigator community. NHBI's strategic vision rests on 4 mission-driven goals that benefit from sustained institute focus and provide a framework for moving heart, lung, blood, and sleep science forward. The 4 goals are to understand human biology. I should advance my slides. To reduce human disease, to develop workforce and the resources, and the last, to advance translational research. NHBI's strategic vision has 8 overarching objectives that organize over 130 research priorities. You can see these 8 objectives from this slide. In the interest of time, I'm not going to get the details, but you can find the information at NHBI website. So soon after the launch of the NHBI strategic visioning document, the Division of Cardiovascular Sciences set up 6 strategic vision implementation work group to address social determinant of health, cardiovascular health, and health inequities, to enhance resilience, to promote cardiovascular health, and preventing cardiovascular disease across the lifespan, to eliminate hypertension-related cardiovascular disease, to reduce the burden of heart failure, and to prevent vascular dementia. So we apply a populomic approach to drive NHBI mission. That is to examine health influence across multiple levels, including biological, lifestyle, behavioral, environmental, social, cultural factors, using multiple disciplinary methods to investigate the web of influence impacting health. Cohort studies are the foundation for us to apply the populomic concept. The epidemiology branch within the Division of Cardiovascular Sciences currently manages a dozen of large population-based cohort studies listed on this slide, as you can see, including cardio, Jackson Heart, and Framingham, etc. And the Hispanic community health study, the study of Latinos, is the one solely focused on the participant of Hispanic origin. So this slide shows the diversity in sex and the race of all our cohorts. As you can see that Hispanic population is fairly well represented. Also, there's still room for us to improve. So Dr. Greg Talavera just presented the SOUL study earlier at this session, so I'm not going to get into the details anymore, but I think just one point is the SOUL study is really one of the largest investment from NHBI. As you know, when we create a cohort study, we typically follow the cohort for a long time to come, unless really something goes wrong, terribly wrong. So we not only conduct repeated measurement of disease risk factors typically every 5-6 years, but also track the long-term health outcomes for this cohort. The Hispanic community health study, the study of Latinos, started the recruitment in 2006. Now we are in the third clinical examination phase. The Hispanic community health study, so the study of Latinos can potentially provide opportunities to explore cancer-related ancillary studies. It is worth mentioning that we also found the SOUL youth study. However, young age and the limited number of sample size may be a challenge to conduct cancer-related studies for the time being. We also encouraged several other cohort studies to include participation of Hispanics. For example, we support the Omni-1 and Omni-2 cohort through the contract to the Framing Heart study. Omni-1 and Omni-2 cohort include Hispanics in the Boston area. The last but not least point I'm trying to make is about the trans-NHBI and the trans-NIH collaboration and the data sharing platform NHBI has developed. One example is Transomic for precision medicine, or the TopMed study. TopMed is part of a broad NIH-wide precision medicine initiative, which aims to provide disease treatment tailored to an individual's unique gene and the environment. TopMed contributes to this initiative through the integration of whole genome sequencing and the other omics data with molecular, behavioral imaging, environment, and clinical data. We use TopMed, we use the bio data catalyst platform to share data among research investigator community. We have tried very hard to include the participants with geographical, racial, ethnic, and genetic diversity in the TopMed study. We are not there yet as far as Hispanic participants are concerned, but 15% is a good starting point. In closing, just want to point out that the cohort studies we manage at NHBI may serve useful resources for cancer-related studies. We look forward to having more collaborations with the cancer research investigators community. Thank you for listening, and happy to take any questions you may have.
Thank you very much, Dr. Hong.
Thank you.
So thank you to all the speakers. We have heard 6 excellent presentations on different topics. We have very limited time remaining, so I would like us to get straight into the questions so that if there's any burning issue that we need clarification on, we'll try to get those answered. So, when you look at the chat, it looks like our speakers did a marvelous job because not much comments in the chat, but I'd like to encourage anyone who has a question, don't be shy to raise your hand and we'll try to get your question. I do have a couple of questions that came through. So the first one is for Dr. Wendy Setiawan. And the question is that given that the multi-ethnic cohort study has been leveraged across many published studies, as you know, that can you comment on the generalizability of the findings to other groups of Hispanic heritage?
Right. So as most of people know, the MAC Hispanics, they're mainly from LA County. And they recruited in the early 1990s. And during the recruitment process and during the baseline characteristics determinations, what we found is the Hispanics populations, in terms of looking at their education level, SES, they look similar to what we observe in California, in Southern California. So that's like one thing that we feel confident in terms of generalizability of the MAC Latinos for California Latinos. But obviously, we are aware there's widely difference in terms of, Latinos in California versus in Texas and Florida. So whenever we write papers or we write grants, we always have to be aware that, there's limitations in terms of generalizability and people should be aware of in terms of interpreting the results.
Thank you very much, Wendy. The next question is for Dr. Raul Cateno. Hope I'm pronouncing it correctly.
Right.
So the question is that alcohol consumption, especially at high levels of drinking, is associated with increased risk for certain outcomes in terms of the conference cancer. And we have heard from Dr. Greg Calavera on cardiovascular diseases. So in terms of the national surveys that you have conducted, can you comment on the, you didn't talk much about your findings. Can you comment on the findings in terms of the type of alcohol that is consumed, type, frequency, and dose of alcohol consumed in your population, in the Hispanic populations, probably by, the different groups in that population?
Yeah, in the United States, most Hispanics drink beer. It's the preferred beverage for Hispanics and for many other groups. There is some liquor drinking in the United States, but it's not compared, let's say, to countries like, Russia and other Eastern European countries where vodka is the preferred beverage. And so Hispanics are mostly beer drinkers in the country. What was the other part of the question, please?
In other than type of alcohol, can you comment on the frequency of use and possibly the level of drinking or the dose?
Yeah. If you compare the usual comparisons between Hispanics and, let's say, the white majority of the population, what they show is that there is a slightly elevated frequency of binge drinking among Hispanics. And binge drinking is defined by the NIAAA as 5 drinks in about 2 hours for men and 4 drinks for women. And a drink is about 1.5 ounces of spirits, a 5-ounce glass of wine, and a 12-ounce can of beer. Hispanics have that type of drinking a little bit more elevated than whites. If you look at Hispanics as a group, there are considerable differences across national origin because Puerto Ricans and Cuban and Mexican Americans drink more than Cuban Americans and South Central Americans, including binge drinking.
Thank you very much for that clarification. So another question here for all the speakers, and this is in light of the talks by Drs. Greg, Raul, Wendy. Some of them are not focused on cancer, of course. And also in terms of the talks by our NIH colleagues, the question here is, could maybe one or two individuals talk about or comment on the steps forward to expand engagement across NIH collaboration to support cancer research in Hispanic populations?
I can go first and report that we just funded a small project, a young investigator, early career investigator, to link our sample with the cancer registries, which I think will improve. And if that can be done and done so accurately, that will provide a rich data set as well because of the well-characterized sample that we have. So that's one thing we did.
I can also answer that question some that, like acknowledging that the MAC is an aging cohort. So we've been actively work with NIAA, the Aging Institute to identify the areas of research that's really important that we can address in the MAC. And then one of the examples that we've been successful is for the Alzheimer study, where we receive supplement to expand beyond cancer research, looking at the determinants, looking at genetics, biomarkers, and lifestyle factors for Alzheimer risks in multiple populations. So that's what we've been doing in the MAC, among other things.
So I could speak on that from the funders' point of view. I think this meeting is the first good step that we're doing. So the fact that we're all here and we're presenting shows that there's a commitment. But as Wendy stated, NIA works a lot with the other institutes to really look for opportunities to increase AD studies in minoritized populations. And I know the question was really about cancer, but I think what we need to do if we're going to work across institutes is that we really have to take a more comprehensive approach of looking at multiple disease and multiple chronic condition outcomes. One of the things that stands out to me, being a former program director at the NCI and now working at NIA, is that the challenges are very much the same. Just like cancer, AD is multifactorial in nature. There's a larger prevalence of AD in our minoritized populations, yet these are the populations that we study the least. And so there's a lot of room for us to really start to work together as institutes to see how we could increase numbers overall. The Unite initiative may present a perfect opportunity for us to do that. And then there's other interests specific to NIA. NIA has an interest now in looking at research related to dementia and cancer. And so there may be opportunities for us to work with NCI down the line. But I think a more comprehensive approach is the way to go, especially if you're going to try to get the interest of other NIH ICs.
Thank you very much. In the interest of time, our time is counting down rapidly here, but we just want to get this other question in. So we have heard from outside researchers with resources like the multi-ethnic cohort study, like Dr. Talavera's studies and Dr. Catino's studies. So the question is here, for researchers who are not directly associated with these studies as investigators or part of the same institutions, how is it that people who are not connected directly can get access to these resources, especially researchers who are of Hispanic origins?
I can go first again.
Please.
On our website, if you go to our website, we have a tab there for that basically says how to get involved in the Hispanic Community Health Study. And the content within there will direct you to our career development meeting that meets monthly. We have special interest groups where you can participate openly in areas that are looking at cardiometabolic or gastric issues and things like that. There's over 10 of them. That's one way. And then we have regular meetings of diverse groups related to our ancillary studies as well, because not only are we funded by NHLBI, but many of the other institutes sponsor our ancillary studies. So I'll stop there.
Just
2
want to
minutes
add,
remaining, please go ahead.
just want to add that Greg has said, I think most of the NHBI's sponsored cohort study, for example, Framingham, you can literally go through our website. You can have access to the data. Of course, when you propose any ancillary study proposals, you have to go to the executive committee of that study. But I think it'll be good that involve one of the investigators from that study. But yeah, you can have access on our website. And you can go to the, for example, Framingham, their own website, but they will charge you some fees. But for NHBR website, it's free.
I think another approach that we could use too is think about training people who don't usually do health disparities research to do disparities research. And this is something that NIA has been doing a lot through the Butler Williams Scholar Program. We bring in very talented researchers that are doing great research, and these are junior investigators that are doing great research, but no disparities. And so we really try to connect them with disparities researchers. We review their specific aims. We see how they could add disparities component to their specific aims. And this has really been successful in increasing the number of our junior investigators and early stage investigators in doing disparities research. We found that our community is very open to data sharing. Hopefully with the new policies coming down the line in 2023, that would improve the will and and the ability of our investigators to share their data, particularly for disparities research.
So thank you all. Last 1 minute remaining, so go for it.
I like to add for the MAC, we have a specific T32 that's funded by NCI that focuses on training postdocs to focus on disparities research. So that's just expanded our ability to train future investigators to focus on this.
I'm going to just ask one more quick thing here. This is for Dr. Rina Das. A question came for you, so just maybe this is a short answer. Is there any funding opportunities for international students as postdoctoral fellows within the programs that you have mentioned?
Well, we have the traditional postdoctoral mechanisms that we fund. These are the career development funding announcements and the fellowship funding announcements. So we follow those guidelines. If they allow for US citizens or some international, then we follow that. But traditionally, we focus on mainly US citizens in the programs that we fund.
So I'd like to end this session by thanking all of you for your excellent presentations and also doing a marvelous job in answering the questions. I think you have added a lot to our conference today. So thanks a lot, and hopefully we'll be in touch in the near future. Take care.